We should start on time, so it's already 2. I'm Didier THARREAU from CIRAD, France, and I have the pleasure to co-share this session with Kateryna UDOVYCHENKO.
Yeah, correct.
from Ukraine. Just before we start, just a brief introduction how this particular session came up. Of course, you all know that on February 24, 2022, there was a military invasion of Ukraine by Russia. And following this dramatic event, Professor Alex Shevchenko proposed to the organizing committee to have a particular session on plant pathology and food safety in Ukraine. The scientific committee acknowledged the importance of such a session and supported this proposition, but also wanted to enlarge this proposition to broaden it to other geographic areas where, because of wars and conflicts, such wars and conflicts are impacting research in plant pathology and food safety. So we know that it's the case in many countries for many years. So we wanted to be to associate other countries to this session. So I want to take this opportunity to thank the scientific committee for really having done a great job, not only on this session, but for all the meeting, and I hope you will appreciate the work they have been doing. It was already mentioned that ISPP has been very active in helping people and by, in particular, funding some resilience bursaries for plant pathologists. And I want to mention also that the Polish Phytopathological Society was very active in welcoming Ukrainian colleagues. And that there were also other bursaries for Turkish colleagues and for help for Syria. And okay, so I won't mention all the societies who helped, but which helped and supported this, but there were several. And so this session is part of these actions around conflicts and wars, and to especially keep us aware of problems of our colleagues and on the consequences of such dramatic events in the world. So I won't be longer, and we can start the session right now.
So, dear colleagues, we'll start our session from talk Research and Education in Plant Virology in Ukraine. The present is foggy, the future is bright, by Irena BUDZANIVSKA from Taras Shevchenko National University of Kyiv. So, please, Irena.
Are you hearing me? Oh, yeah. Okay. Dear Chair, dear colleagues, first of all, I would like to express my sincere gratitude to Organizing Committee for the opportunity to participate in the Congress. I would also like to thank you for highlighting the impact of war on research and education in phytopathology and food security in countries. Today, I stand before you to share my thoughts on the past, present, and future of plant virology research and education in Ukraine. My country has not only a rich history and diverse culture, but also deepened root connection to agriculture. Since the times of Tripilla and Kievan Rus, my people have been engaged in the cultivation of agricultural product. Ukraine, often referred to today as the breadbasket of Europe, is a true agricultural paradise with these fertile soils and vast expanses of arable lands. Located in Eastern Europe, Ukraine's agricultural heritage goes back centuries, making it an agricultural country known for its bountiful harvest. From golden wheat's field stretching as far as I can see to sun-drenched orchards, Ukraine's landscape are an artist's dream and a testament to the nation's agricultural prowess. Ukraine's 4 climatic zones allow for the cultivation of a wide variety of crops, grains, legumes, potatoes, vegetables, sunflowers, fruit, berries, and others. Agriculture is closely intertwined with Ukraine's identity, shaping its economy, culture, and traditions. With such a deep agricultural heritage, it is not surprising that Ukraine's scientific community is studying plant viruses. Preserving crops and ensuring food security for the country and beyond has become an important task for Ukrainian scientists. However, the deep connection to the land has created challenges that go beyond the natural seasons. Since February 2022, a year and a half ago, a shadow has fallen over our aspiration. The shadow of war and uncertainty that has come to our country. In my speech, I will discuss the impact of the conflict on plant virology research and education in my country. So, some history facts. Everyone knows the founder of virology is Dmitry Ivanovsky, who researched new diseases on tobacco and published his work 1892. However, few people know that he did his work in the south of Ukraine, where at the time many tobacco growers were engaged in growing tobacco for export. Even less well known is the fact that in 1903 at St. Volodymyr University in Kyiv, Dmitry Ivanovsky defended the world's first doctoral dissertation in virology, devoted to the properties of the pathogen he discovered. At the university, which now called Taras Shevchenko National University of Kyiv, where I am honored to work. It is with immense pride and gratitude that I share this unique facet of our natural history. So, the cradle of virology is Ukraine. I am very proud of this. Moving forward to the present, it's imperative to acknowledge the widespread presence of scientific and educational institutions dedicated to research and training in the field of plant viruses across all regions of Ukraine. As highlighted by the map, these institutions are distributed across the country, from Kyiv, Lviv, Zhytomyr, Kharkiv, Odessa, and Crimea. If we turn our attention on the slide, you will find a list of universities and research institutes are leading on plant viruses research. For example, Kyiv National University, another university from different towns, different parts of Ukraine, and scientific institutions. For example, the Bolotny Institute of Microbiology and Virology and Horticulture Insitute. We conducted a meta-analysis by searching Scopus, Google Scholar, and Web of Science databases using the keywords plant viruses in combination with affiliation with Ukraine. As shown on the slide, it is clear that the number of publications in this field has been growing markedly since 1991. This upward trend emphasizes the growing interest and involvement of Ukrainian researchers in study of plant viruses. Even in comparison to 2021, it's remarkable to observe that the number of publications continue to rise. This trend highlighting the resilience and determination of Ukrainian researchers who, despite the challenging circumstances, are contributing actively to the field of plant virology. Displaying on this map is the distribution of publication actively across different regions of Ukraine. What stands out in the notable trend where the result consistently show higher publication activity in regions with a comparatively low number of affiliated institutions. This intriguing observation challenges conventional expectation and suggests the factor beyond sheer numbers play a significant role in driving research output. It underscores the potential impact of focused effort, collaborators, and perhaps unique local strengths that are contributing to this dynamic pattern. The numbers we have seen a facet of our scientific engagement, but they don't tell the whole story. Beyond the statistic, there lie a narrative of resilience, challenges, and adaptivity that researchers and educators have been navigating amidst these turbulent times. While publication activity has demonstrated an upward trajectory, we must ask, what factors are influencing this trend? How is the war altering the landscape in which our scientific endeavors unfold? Here, some of the factors that I believe have had an important impact on plant virology research and education in Ukraine. Broken research and collaboration links, brain drain of Ukrainian scientists, relocation of university and academic mobility abroad, destruction of plant pest control system. Let us take a closer look at them. The conflict in Ukraine has severely disrupted research and collaboration ties among Ukrainian scientists. Institutions located in conflict-affected regions have faced challenges in maintaining normal research operation, hindering the progress of important plant virology studies. Communication barriers and infrastructural damages have further limited the sharing of expertise and scientific knowledge within the scientific community. To overcome these challenges, Ukrainian scientists have resorted to innovative solutions. They have increasingly turned to virtual communication platforms such as video conferencing and online collaboration tools to stay connected with colleagues and share their research findings. Additionally, international collaboration and partnership have been fostered to ensure community in research and educational activities. This global connection have enhanced researchers to access resources, data, and experience from abroad. The instability caused by the war led to brain drain of talented Ukrainian scientists in the field of plant virology. Many experienced researchers have been compelled to seek safer options and better research opportunity abroad, resulting in a significant loss of expertise and knowledge within Ukraine. To mitigate the brain drain, concentrated efforts are being made to create attractive incentives for scientists to remain in Ukraine. These incentives include offering competitive scholarship and research grant, facilitating access to cutting-edge research infrastructure, and promoting a supportive research environment. By investing in the career development and well-being of scientists, Ukraine seeks to retain and attract top talented in the field of plant virology. The conflict has led to the relocation of several universities from conflict-affected regions to safer areas within the country. Furthermore, many students have opted for academic mobility programs abroad, leaving the country's educational institutions with a reduced enrollment in plant virology program. Ukrainian university and the government are actively supporting the continuation of educational program and plant virology in regions that remain unaffected by the conflict. Efforts are being made to encourage online and distance learning initiatives, allow students to pursue their studies remotely despite the challenges posed by the conflict. This initiative aims to maintain the quality and accessibility of plant virology education for interested students within Ukraine. The war has resulted in the destruction of some plant pest control systems in Ukraine, making it challenging for authorities to effectively monitor and control the spread of plant diseases. This poses a significant threat to agricultural productivity and food security in the country. In response to the destruction of plant pest control system, international organizations and partner countries are stepping in to provide support to Ukraine. They are extending assistance in enhancing the country's capacity for monitoring and controlling plant diseases. These include providing technological assistance, training, and sharing best practice in plant health management. Envision the future of education and research in plant virology, a future of prosperity, collaboration, and excellence. Through dedication, international partnership, and support, this bright future can be actualized, advancing science and fortifying Ukraine's agricultural sectors. Confirmation that scientists and educators in Ukraine are actively engaged is evidenced by the upcoming 10th International Conference Bioresources and Viruses, scheduled to be held in Kyiv in September of this year. This conference, initiated in 1994, served as a testament to our dedication to advancing knowledge. The event will encompass research from various fields of virology, with particular emphasis on studies concerning plant viruses. This year's conference holds special significance, gathering researchers from around the globe. This conference underscores our commitment to collaborative research, knowledge dissemination, and the pursuit of solutions in the realm of virology. It serves as a reminder that even in the face of challenges, the spirit of scientific inquiry remains resolved. Peace. In my greatest desire, peace in my soul, my family, my country, peace in the whole world. If we have peace in our world, we can overcome any challenges that lie ahead. Today, our understanding of viruses has evolved. We can not only diagnose them, but also predict the course of diseases and anticipate harvest losses. However, we need to go further. The interaction between viruses, their vectors, and plants in a complex mechanism that requires a deep understanding. Knowledge of this interaction will ensure the sustainable function of the ecosystem. After the war, it is possible that first international center dedicated to studying plant viruses as equal participant in ecosystem will be established in Ukraine. Our knowledge of viruses is profound, yet many questions remain unanswered. Today, I want to express my gratitude to all of you for your support, compassion, and assistance in our struggle. I also want to extend my heartfelt thanks to my colleagues working abroad who contribute to the development of our country. However, my deepest gratitude goes to those who are currently living and working in Ukraine. It is who, through your diligence, are brightening up closer to victory. Peace in Ukraine and through the world is our shared goals, and only together we can achieve it. Thank you for your attention, and Slava Ukraini!
There is some problem with the application, so if you ask question, maybe it did not reach us. All the questions from the session closed by and ours are coming to the same place, so we don't know. So we will use a regular way. If you have questions, please raise your hand.
Thank you for your presentation. I was wondering, even in the most dire circumstances of Ukraine at the moment, whether there were learnings in agriculture through forced change in practice, like not being able to apply the same traditional, applications of pesticides or whatever, in terms of sort of more organic forms of pest control, natural suppression through an inability to apply what would be conventional means.
I am not sure that I truly understand, but I think now in Ukraine farmers who work now, it's very actively, use usual practice and produce not only classic foods, but organic foods too. Катя, я правильно зрозуміла? Yeah?
I would like to thank you. And I would like to ask, what do you consider as the main virus problem? It's a very practical kind of question under the circumstances. It may actually have changed in the meantime, but what is?
I think at that time we have a main problem with cereal viruses.
This?
Cereal
Virus.
Yeah, it's the main problem in Ukraine now. But we try to understand this problem and try to solve this. Mm-hmm.
Mm-hmm. Thank you. A success.
Just a comment, maybe not a question. You have mentioned, thank you very much for your talk. It was very interesting, and overall it showed the situation in Ukraine. I come from Poland, and many researchers are at present in Poland. And I would like to say that this opened the science. Of course, war is the evil, but the relocation of many researchers also opened the science in Ukraine in a way, and we are very grateful that many researchers from Ukraine work with us. And I would like to stress that when we publish, we always show the origin of the institution of this researcher, this particular researcher, so it can be either the Ukrainian institution or just the double affiliation if Polish or international scientists give funding. So this may be one of the answers to your very big bar in 2022, 2023,
Maybe,
that
maybe.
many researchers from Ukraine work all over, and if they keep their original affiliation, this adds to your science as well.
Yeah.
So thank you
Yes.
very much.
Thank you.
And the next speaker is Alina DUNICH. Impact of war and climate change on viral disease of winter wheat, threat to food security. She is from Ukraine, and fortunately this report is pre-recorded, and Didier will start it now. Yeah.
Dear participants, let me introduce my presentation about impact of war and climate change on viral disease of winter wheat and threat to food security. Ukraine is one of the 5 largest exporters of grain on the world market, supplying more than 24 million tons of wheat grain to the world market every year, especially to countries with food shortages. Several countries in Africa and the Middle East are largely dependent on imports of wheat and other agricultural crops from Ukraine. Monitoring and control of plant pathogens are of strategic importance for the bio and food state security, sustainable production of plant products for human and animal food. Underestimation and lack of knowledge about viruses, as well as the emergence of their new variants, is very dangerous and can lead to the global economic crisis and unpredictable consequences. For example, at least 10% of global food production is lost to plant diseases, with an economic value of $220 billion annually. Plant viruses should also not be underestimated, as they are a significant economic factor and cause a significant reduction in crop yields. Unlike human and animal viruses, for which drugs and vaccines can be made working and learning remotely, viral plant diseases pose a threat to food security because there are no effective protection methods for them. Our long-term research showed that in Ukraine are circulating 3 viruses: wheat streak mosaic virus, barley yellow dwarf virus, and wheat dwarf virus. And these viruses have high epidemic potential because lead to decrease in wheat yield. Wheat streak mosaic virus causes typical symptoms of streak mosaic on the wheats. This virus was detected in all southern regions of Ukraine. It was found that virus-infected wheat plants differed significantly from healthy ones in such parameters as spike lengths, here is infected and here is healthy one spikes from healthy plants, in such parameters as number of seeds per spike, weight of seeds per spike, and this mosaic virus is decreasing protein content in infected plants. Phylogenetic studies of wheat-streak mosaic isolates showed that they are belonging to the clade B, as are the wheat isolates originated from Europe. Besides, 2 isolates sequenced by us in 2018 has 12 and 25 amino acid substitutions in the capsid protein region. It is interesting that most of these amino acid substitutions are located in the same positions as in isolates of other clade named as B1 clade. B1 clade includes non-crop, non-wheat isolates. These substitutions revealed in Ukrainian isolates were not found in other foreign European isolates of wheat streak mosaic virus. So, Mutations in nucleotide and amino acid sequences of capsid protein gene of wheat streak mosaic virus can lead to changes in their properties or in their virulence, and thus increase their epidemiological potential. In 2020, a wheat dwarf virus was detected by us in 14 wheat cultivars in 5 regions of Ukraine. The plants exhibiting different symptoms, including dwarfism of the plants, no heading, and yellowing on the leaves. Our research has shown that the wheat dwarf virus has a significant negative impact on wheat productivity and wheat yield, because in many varieties it leads to the reduction in spikes and other parameters. Our observations revealed several impacts of viral infection on plant productivity. In general, no heading of plants was more than 50%, and in some areas up to 100%. In virus-infected plants that formed spikes, various degrees of spike damage was also observed. The spikes looked too weak comparing to the healthy ones. They contained fewer grains, and their grains were smaller in size than healthy. On phylogenetic tree, the studied isolates is located somewhat separately. It can be explained by different intensity and selection pressure, resulting in appearance unique amino acid substitutions that we have revealed in their capsid protein gene. The next our step was to analyze the rate of synonymous substitution per synonymous site and the rate of non-synonymous substitution per non-synonymous site. The larger difference between these parameters, the larger is selection pressure. So it was found that the global ratio dN and ds was founded for all isolate sequences and indicate negative selection due to functional and structural constraints which occurs in proteins having attained a high level of adaptation. But for Ukrainian isolate from Khmelnytsky region, this indicator was the highest. Our results revealed that this isolate from Khmelnytsky region more significant reduced the weight of seeds per spike and the weight of 1,000 grains than other isolates from Kyiv region. This fact can be explained by more mutations in capsid protein gene of such isolates compared to the Kyiv isolate. So this suggests that Khmelnytsky isolate has higher potential divergence, as its sequence contain more non-synonymous changes, which are subjected to selection, as they can alter the protein function or structure, for example, because even few changes in the nucleotide sequence can be a big impact on the virus functionality, as has been demonstrated, for example, for other viruses, for maize streak virus. It is known that the composition of a wheat viruses depends on the weather conditions that occur of crop sowing and during the growing season. Manifestations of viral diseases can be observed annually under any weather conditions, but outbreaks occur periodically because of massive reproduction of insects, vectors, and their prolonged nutrition on plants under conditions of warm, prolonged autumn periods. Sans barrel yellow drought virus and wheat streak mosaic virus are transmitted by vectors, by aphids and microscopic mites. Climate change can indirectly affect the intensity and prevalence by affecting the reproduction or overwintering of their vectors. In this regard, we analyzed the dependence of virus circulation on agroclimatic conditions in agrocenosis of some regions of Ukraine. The main vector of its mosaic virus is the wheat cormite, and for viral yellow drought virus, it is several aphid species. It is known that dynamics of increasing aphids and mites number is determined predominantly by hydrothermal conditions, which significantly affect the cycle of insects' development. Cool and rainy weather in summer limits the propagation of aphids, and the hot and dry weather contributes to their early appearance on plants. Thus, air temperature and rainfall data was analyzed in our study. Meteorological monitoring data showed that in 2011, 2013 and 2015 frequency of barley yellow drought virus was on a lower level, as it correlated with weather conditions. So in March and in June, which are potentially favorable months for the aphid propagation, in these months, it was significant increase in the rainfall amount was noted by us. June and March. Frequency and intensity of rainfall influence the barley yellow drought virus epidemiology. Consequently, in years with more arid spring and more arid summer, for example, 2012 and 2014, barley yellow drought virus prevalated in these years. One of the stages of life cycle vectors of wheat mosaic virus and barley yellow drought virus is the overwintering on wheat or winter crops of various perennial grasses, and we had also analyzed such parameters as temperature at soil surface, temperature on terminal node depths of winter wheats, and maximum soil freezing depths in winter. And it was established that under more severe winter conditions in comparison to previous years, barley yellow drought virus circulates, and wheat mosaic virus under severe winter conditions is almost not detected. For example, in 2012, the average monthly temperature in February was much lower in comparison with previous year. So average monthly temperature at soil surface and minimum soil temperature on terminal node depths of winter crops were significantly lower in comparison with previous year. This may be due to the fact that such conditions are unfavorable to the mite with cormite vector of its mosaic virus. In contrast, in 2011, 2013 and 2015 years, when wheat mosaic virus was detected, winter was significantly warmer and shorter than in previous year. Such weather conditions can contribute to the reproduction of wheat mosaic virus vector, named as CIRAD-19. Besides that, during our research, we noticed plants that had symptoms of yellowing and reddening of the leaves, which are quite similar to the symptoms caused, for example, by barrel yellow dot virus or wheat streak mosaic virus. And here is the plants with reddening, yellowing on the leaves. Yeah, and here is in the figure 1 you can see the symptoms caused by barrel yellow dot virus, and these symptoms is very similar as you can see. But in such plants with reddening and yellowing of the leaves, there are no viruses was detected. So we have started to analyze average daily temperatures in May in these fields. And we have revealed that high positive temperatures during the day were replaced by low temperatures at night, and the temperature difference during the months was 19 degrees. Such a temperature difference leads to change in carbohydrate balance, which arise as a result of non-specific reactions of plants to stress. In wheat plants, under the influence of cold stress, there is an increasing synthesis of anthocyanins, which is manifested by external symptoms, for example, a change in the color of the leaves from the normal green to red. And interesting that such reaction is similar both to stress and viral damage. And that's why virological studies are extremely important to establish a reliable reason of the appearance of such symptoms, and will contribute to reducing the use of chemical plant protection agents and environmental pollution. Even prior the war, global supplies of key staples were tight, and in stocks of wheat and other crops such as maize and soybeans were at their lowest levels for many years, and prices were high compared to 2020 levels during effects of the COVID-19 pandemic. In 2022, this situation became more complicated as a result of war waged by Russia against Ukraine. Global supply chains of food products from Ukraine were disrupted. Tons of grain stolen by Russia from occupied territories, which led to deficit and high prices for agricultural products in the world. When Russian troops invaded Ukraine 1 year ago, the war appeared to pose a grave threat to global food security. Ukraine ships almost 75% of its agricultural exports through their ports of Black Sea. And when war was started, Ukraine found its Black Sea ports effectively blockaded, sharply limited its ability to export its 2021 crops, and the planting and harvest of our 2022 crops disrupted. Within a week of invasion, prices of wheat futures had jumped almost 60%. Later, the partial reopening of ports through Black Sea Grain Initiative helped it facilitate additional exports from Ukraine, and prices on wheat had fallen back almost to pre-war levels. But unfortunately, from 18th of July 2023, the Sea Grain Initiative was stopped by Russia. As a result, in addition to the decrease in crop yield due to damage by viruses, due to damage in climate change, the following problems arise in today's wartime realities. And the first problem is decrease in sown areas in Ukraine. Decrease in sown areas in Ukraine due to active hostilities, occupied territories and mined fields. For example, for today, decreasing in sown areas in Ukraine is by 45% compared to 2021, and decreasing by 22% compared to the previous year. Decreasing of sown areas is observed in regions without active as fatalities, unfortunately. And the reason for this is a limited export possibility for today, expensive logistics, and the increase in fuel prices. And the second reason is a minefield that we can cultivate for today. Yeah? And therefore, these fields for today serve as a source for plants that can be infected by viruses. And these fields can serve as source for insects that are potential vectors for viruses. And options for increasing food security in Ukraine, the main goal in this situation should be creation of wheat varieties with complex resistance. These varieties must be resistant to viruses, to fungi, and to abiotic factors. United cooperation of virologists, plant pathologists, genetics, and breeders is needed to fulfill this important task. Here is our context in the slides. We are open to cooperation. And thank you very much for your attention.
So there is no possibility to ask, you can ask questions, but you will not get the answers. So you have to ask the questions directly to Alina. So the next speaker is Kateryna.
First of all, I would like to thank to the International Society for Plant Pathology and the organizing committee of the 12th International Congress of Plant Pathology for their initiative and great opportunity to participate in the Congress and present the current situation in horticulture in Ukraine. Here I represent the Institute of Horticulture of the National Academy of Agrarian Sciences of Ukraine, which has been a scientific base for development of Ukrainian horticulture for over 90 years. Ukraine has long traditions in horticulture due to unique soil and climatic conditions favorable for growing of wide range of economically important crops. Especially during the past 20 years, Ukrainian horticulture has been switching to intensive technologies, introducing modern cultivars, high-density plantings, plant protection schemes including bioproducts, special garden constructions, and so on. So the areas of perennial horticultural crops had been decreasing from 2000 till 2015, when they stabilized at the level of 250 to 170,000 hectares. The level of yield per hectare increased at least twice, and also the consumption of level of fruits and berries doubled these years. Now let's look at how the war impacted recent situation in Ukraine. It is already 1 year and 5 months of Russia's full-scale invasion, and the war hasn't only taken many lives, but also severely affected Ukraine's economy and infrastructure, and in particular agricultural sector. The damages and losses suffered by the sector are estimated now by experts of the World Bank, Food and Agriculture Organization, Ministry of Agrarian Policy of Ukraine, Kyiv School of Economics, State Statistics Service of Ukraine, and other professionals, which data are used in my presentation. So, talking about the harm caused by military actions, we should consider a direct damage and indirect losses. Damages could be estimated in terms of the monetary values of tangibles, and now I want to go through the main categories that suffered the most. The first is areas of perennial horticultural crops. And in 2022, its number decreased by 12% across the country comparing to previous year. Due to occupation, the entire area of horticultural crops in Kherson region was unavailable for cultivation. In Donetsk, it was lost almost 60% and 35% of areas in Zaporizhia region. In total, all regions of Ukraine demonstrated reduction of areas because of the direct impact of the war action, economic or other reasons. But considering the decrease in harvesting areas of different crops, the highest losses were for apple as the most popular horticultural crop in Ukraine, and then go sweet cherry and grapes. But in terms of share, the area of sweet cherry orchards was lost the most, around 37%. The biggest orchards of this crop are situated in the Zaporizhia region, and especially in the surroundings of Melitopol city. Due to its unique taste characteristic, Melitopol cherry became the first Ukrainian berry that received geographical indication patent in 2020 from the Ministry of Economic Development of Ukraine. But last year, according to Melitopol mayor, the Melitopol occupation authorities forced large farmers to deliver the entire cherry harvest. And facilities of the biggest cherry producers in this region were generally taken over by unknown persons. The turnover of the Melitopol cherry cluster was estimated at level of $25–35 million per year, and now it is lost for Ukrainian horticulture. Viticulture was another branch that suffered significantly by the war. And according to official statistics, areas of vineyards decreased by 6,000 hectares, which mainly are located in Kherson, Mykolaiv, and Odessa region. The next point is the largest category of agricultural damages, and it is destroyed or partially damaged agricultural machinery and special equipment. It accounts for $4.7 billion and is almost half of the total agricultural war damages. This figure includes machinery which was used in horticultural farm also. And using the baseline of 2019, more than 17% of agricultural machinery and equipment in Ukraine are estimated to be severely damaged and destroyed. Also, many storage facilities or other agricultural premises were totally or partially damaged. On the photo you can see the production and storage facilities of different farms in Ukraine. Enterprise Arctica in the Kharkiv region, which has plantations of berry and vegetable crops and also is engaged in the freezing products. The other photo is the premises of the Bakhmut nursery, which was one of the largest producers of planting material of fruit and berry crops in Ukraine. Besides direct damages, Russia's invasion of Ukraine had indirect effects on the whole agricultural sector that resulted in losses of approximately $31 billion after 1 year of the war. These losses encompass the foregone revenue due to lower quantities of goods produced and additional costs that producers bear due to the war. In case of horticulture, this decrease in crop production is estimated at $429 million, and moreover, farmers who continue their work are faced with extreme growth in production costs. The main reason was the increase in prices for fertilizers, plant protection products, fuel, and other necessary materials and equipment. The growth of costs for fertilizers were from 20 to 100% for some positions. And plant protection products demonstrated an increase of 10 to 20% compared to prior period. Additionally, interrupted logistics chains caused extra cost growth. Also, due to the destabilization of market, were cancelled seasonal credit and installment payments for fertilizers and plant protection products, which were previously provided by suppliers to produce, and as now only total prepayment is available for farmers. The next challenge is shortage of labor. A lot of men are mobilized now to Ukrainian army, and there is also a large number of internal and abroad refugees. As a result, farmers met with shortage or total absence of such specialists as tractor drivers, operators of other machines, hydraulic engineers, seasonal workers, and others. There are also more delayed problems in horticulture, which could be not so obvious yet, but are very important. Due to the military actions, the well-established work of scientific and phytosanitary institutions coordinating work in the field of horticulture was disrupted. In particular, the Institute of Horticulture has a network of 7 research stations located in different regions of the country. Unfortunately, the Melitopol research station is currently under occupation. Its premises were seized by the occupation authorities. Some of the scientists left Ukraine, and those who stayed don't have opportunity to continue their work. Due to its favorable location, this station had a successful breeding program for stone crops, especially sweet and sour cherries. And also it maintained germplasm collections that encountered more than 800 accessions. The situation at the Bakhmut research station is even worse. Fierce fighting continues there today. The premises of the station are destroyed and cannot be restored. Experimental orchards were significantly damaged as a result of hostilities. And this institution was a maintainer of the germplasm collection of sour cherry and niche crops. Despite partial duplication of genetic collections, it is likely that some varieties will need to be restored from private collections or imported from abroad. Another problem is the difficulties in conducting observation and of phytopathogens, both in connection with hostilities in certain territories and indirectly due to the economic reasons. It is important to note that as a result of the increase in the cost of plant protection products, farmers were forced to revise protection schemes, taking into account the shortage of funds. Such an economy in the long run can contribute to the development and spread of pests and diseases of various crops, and it will also become a limiting factor in horticulture. Another important consequence, which we already see today, is the decrease in the production of certified planting material. In Ukraine, as in many other European countries, planting material certification schemes work according to the standards of the European Plant Protection Organization. Therefore, to ensure the high quality of planting material, nurseries conduct additional checks for the absence of harmful pathogens and pests according to the certification schemes. In 2022, we noted a sharp drop in the number of registered nurseries. If in 2021 there were 62, next year it was only 25. And the production of planting material decreased more than 4 times. The amount of imported planting material also dropped significantly. Such a decrease in production is quite expected because part of the nurseries are located in temporarily occupied territories or in war zones, and due to uncertain future, producers don't risk investing in perennial plantations, which has led to a decrease in demand. And the certification procedure itself requires the investment and additional funds. But despite many negative circumstances, Ukrainian horticulture continues to function and seek new opportunities for development. In particular, I would like to cite a number of positive cases. In 2022, government launched grants for the development of horticulture, berry growing, viticulture, and greenhouses. Since the beginning of the program, farmers received already $16 million. After the disruption of logistics routes and the closure of certain export directions, Ukrainian farmers are looking for opportunities to expand export to other European countries and the Middle East. Thus, even in the condition of the war, in the end of 2022, it was possible to reach 85% of the value of the previous years, and the value of the export of apples and berries even increased. In 2022, new plantings also was established on the area of more than 1,000 hectares. Part of the farms, in search of new profitable niches, expanded the assortment of crops and started growing vegetables. For example, this year, God's Farm in Ternopil planted tomatoes and onions. The other company from Vinica, Agrana Fruit Luka, planted pepper, eggplant and onion, and both of them previously specialized only on fruit growing. Some enterprises relocated their production facilities. Due to the occupation of Kakhovka, Chumak, the largest producer of tomato paste in Ukraine, was forced to place its orders at a similar enterprise in Volyn. But now the owners decided to build a new plant in Odessa and are interested in supply with over 1,020 hectares of tomatoes. And last but not the least, a part of industry specialists involved in production and research, who were first to leave Ukraine today, are involved in the work of European firms and research institutions, and hopefully they will return with new experience, ideas, and vision for the recovery of the industry. And in conclusion, I want to thank to all the people and countries that have been supporting Ukraine and Ukrainians. Your words of encouragement, support in project implementation, humanitarian aid, and technical assistance, all of this helps us to move forward and build the future together. I'm confident that our collaboration will enable Ukraine to make significant contribution to global food security and pave the way for a brighter future together. Thank you for your attention.
So, questions for Kateryna?
I'm just curious if the loss of the horticultural infrastructure that you described was due to direct military targeting or as a collateral damage of some nearby region being attacked?
It is both direct and indirect, and these damages and losses are still estimated because it continues growing, but I think most of areas under horticultural perennial crops are in regions not occupied now, but still 12% of areas of horticultural crops are now occupied and lost for because of occupation or war actions and so on.
I can now.
Yeah, please.
No, maybe it's better with the microphone because this session is recorded. Yeah, please.
Thank you very much for the nice presentation. Could you tell a little bit about the diagnostic networks in Ukraine for plant diseases or other relevant factors like soil diagnostic labs and other? Are they operating in regions that are affected or they are not working at this point of time?
Thank you for your question. Yeah, we have state services conducting this research and monitoring of diseases, and also research institutions, and also quarantine stations which check, if something imported. Yeah, all these institutions continue their work on not occupied territories. Yes, but situation now is worse because of mainly economical problems, and also part of specialists were forced to move abroad. So we have problems still, but continue our work.
You mentioned cherry production. But I'm doing a PhD about plumpox virus, Sharka. Yeah. Is it prevalent in cherries? And I was also under the impression that Ukraine was a heavily digitized country. For example, I've seen a lot of digitally locked tractors and a large use of drones. So I was also wondering if there is or was any database on Sharka and how it is spreading throughout the country, or what the sanitary situation is.
Thank you for your question. It is, I'm a virologist actually, and actually a virologist interested in horticultural crops and including plum and other crops. Yeah, Sharka is our favorite virus. Yes, Irina. Yeah, yeah. It is really widespread in Ukraine. I think all regions where it is growing, we can find Sharka in plum gardens actually, and also we conducted research about plum pox virus in cherry. Yes, we can find also, especially last 2 years ago, yeah, I think. Yeah, it was, we found on Mahaleb, Prunus Mahaleb, you know, and also on some other cherry varieties. And I think it is PPVM and PPVD strains. Yeah, we can, yeah. And cherry also, yeah. Yeah, it was in Irina's lab, and PPVM was in our lab, so yeah. It's very interesting topic for us. Yeah. And the question other was about tractors, yeah?
Yeah, digitalization and databases.
About databases, I'm not sure, yeah. Better, like direct communication, we can tell you more. Yes.
Okay. Thank you very much. So the next speaker is Dr. MOUAFO-TCHINDA from Department of Plant Pathology, University of Florida, Gainesville, in the USA, and he will be talking about his experience in Rwanda and Burundi.
Thank you. Hi, everyone. So today we are going to discuss about crop pathogen and pest community and severity in complex landscape. And we are going to focus on the Lake Kivu region of Rwanda and Burundi. So the previous presentation just highlighted the impact of war in agriculture. And we all here know that crisis and natural disaster are two of the greatest threat to global food security. These natural and man-made disasters come with a lack of time for farming, a lack of infrastructure that contribute to improve the food insecurity of people. Like, as you can see in this picture, when there is a crisis somewhere, people move with their goods and also with agricultural commodity that can contribute to the spread of disease around the world. I'm from Cameroon, and now Cameroon is facing many conflicts and crises. Like in far north Cameroon, we have the Boko Haram conflict, and also the separatist crisis in northwest and southwest Cameroon. So what we find in Cameroon is that like people move from their village to another village and just come back during the harvested period to harvest their food and just run away. And those who stay in the village just go to the farm when there is planting material, during the planting period and also the harvested period. So they don't have time to manage their field. So, yes. So usually the most vulnerable people in low-income countries live in complex in complex landscape structure with the mixture of cropping system, large number of crops, and there is a lot of exchange of seed, vectors, and pathogen between growers that contribute to build a melting pot of pests and disease difficult to manage. So, many vulnerable regions around the world, in many vulnerable regions around the world, vegetatively propagated crops like banana, cassava, potato, and sweet potato are key food for food security, nutrition, and income. So they are staple crops, and they produce more food than per area than some other crops. Sweet potato, for example, has a short maturity, 3 to 4 months, and flexible planting date. And sweet potato can produce with just only 2 months of rainfall. And also, just a small piece of sweet potato can contribute to complete the daily vitamin A requirement for a kid, and also provide sufficient vitamin and mineral for kids. And with the current crisis in Ukraine, many countries in Africa was not able to have enough wheat. So in those countries, bakers combine wheat flour with banana, cassava, and sweet potato flour. And that contributes to increase the pressure on these crops. So, despite the high value of this crop, the pressure of pests and disease in this location are increasing, and that contributes to reduce to low the production and increase the food insecurity in those areas. Here my colleague Thomas Sharma highlight the importance of seed degeneration in potato, because usually growers in those locations use their own seed, and usually, like after many cycles, there is an accumulation of disease which contributes to reduce the production yield. So here, the main question is, how can we help people in vulnerable location to be able to produce, taking into account the lack of time for farming and the lack of infrastructure. So in this work, we are going to focus on the region of the Lake Kivu region of Rwanda and Burundi, and we are going to identify the important location for pathogen and pest spread using cropland connectivity, and we are going to characterize the pathogen and pest community in those in that location. And we are going to evaluate the relationship between climate, host landscape structure, and disease. And we are compare the machine learning, many machine learning models. So, as I was saying, we are going to focus on the Lake Kivu region of Rwanda. So the Lake Kivu is one of the greatest lake in one of the Africa's great lake in East Africa. So, and the Lake Kivu border like Democratic Republic of Congo, Rwanda, and Burundi. And this region is really vulnerable because there's a lot of crisis here, and rebels always there's a lot of crisis there, and a lot of rebels that contribute to increase the vulnerability of growers in that region. So, Like I was saying, we are going to start this work with host connectivity. So we use crop of banana, potato, sweet potato, and cassava data from MapSpan 2017, and we calculate the relative likelihood of movement of pests and disease at cross location. So in this first map, the number represents the node. It can be like the village or the field. So as you can see here, the number 70 represents the high density location, And here we have the network of host connectivity. So here the link represents the likelihood of pathogen movement between nodes. And this map represents the map of host connectivity risk index. Here the host represents the location heavily connected, and the bridge represents the location connecting region that otherwise will not be connected. So here is our result. So the top maps represent the host density of banana, cassava, potato, and sweet potato, and the bottom one represents the host connectivity. So with this analysis, we were able to identify the most important location for disease establishment and disease spread, because cropland connectivity helps us to understand the most important location for the disease, the movement of pests and disease. So we also collect data in many villages and fields in the Lake Kivu region, in Rwanda and Burundi. We had at least 200 of fields, and the data we collected was were the number of pests and pathogen, incidence, severity, and infestation rate. So after this, we were able to have the community, the pest and pathogen community in those areas. And here we have the major pests and pathogen in those locations. Here with banana, cassava, potato, and sweet potato. Sorry. Okay. This map represents the incidence of pests and of those pests and pathogens. And we did it during the dry period in purple and rainy period in yellow. And we have this one from Burundi and this other one from Rwanda. So we were able to identify like the most important disease or pest and disease, like we have sweet potato viruses and also potato viruses. And here we have Rastonia. And we also find that some pests and pathogens like sweet potato butterfly and sweet potato aphid was absent in Rwanda but present in Burundi. Yes, so here is the incidence, and because we put the rainy, the dry incidence on rainy data, that's why we have two incidents here. So, yes. So after this, we used data collected in this study with to run machine learning model. So the idea here was to see how we can use like landscape data, weather data from NASA power, and also data pest and disease information to build those machine learning models and help growers to be able to set up the good timing for pest and disease management. So we run all the models and we were able, and we compared the performance of different models, and we found that super vector machine and k nearest neighbors random forest had the best performance. So the performance of model was good for some pathogen and pests like Phytophthora infestan and leaf minor fly, but poor for the other like Rastonia. And we also find that the importance of different variables was different from pest and pathogen to another. Like with leaf minor fly, temperature, host density, and host connectivity were the most important variable, but with Phytophthora infestan, relative humidity, precipitation, and temperature was the good one, the best one. So, at the end of this study, we were able to identify the most important location to monitor against pest and disease establishment and spread in those locations. And we were able to, we also found that the pest and pathogen profile was different in each country across season, and weather variable and geographic structure of crowdland can be used to predict pest and pathogen severity in vulnerable location. Like, of course, we think that more data are needed to develop on-farm decision support model, because this one was like the first step, and we are looking forward to go more deeper with this analysis. So I would like to thank my colleague and all of our founders. So thank you, everyone.
Doing some sport. I have one question. I have one question here. Here. On your right side. Sorry. Just to start the discussion. As far as I understood, you are looking at measuring connectivity based on the density of plants. Okay, that's to determine the points where you will be doing surveys because you consider there is high risk of emergence of the disease. Is that correct? I was thinking of that. There is one parameter that maybe you do not take into account is movement of seeds or material by people. But that's okay. That's long distance migration and sometimes single events or not frequent events. Do you integrate that, on how do you take this into account?
Like, first, we were using top-line connectivity first to identify the most important location, because that is like one parameter. If we take like one parameter, we didn't do it to identify where we are going to do the survey.
Okay.
So we just try to map and identify the most important location that decision maker in the country will be able to focus there because they are really vulnerable. And also we did the survey to be able to capture like the main crop and disease, and also come out with some advice. And in our lab, we are doing a lot of trade network, and I have a poster there, I think P31021, yes, with trade network. So we are doing a lot of trade network, and we are, of course, we don't have the trade of seed here, but I also have a lot of work that I'm running now with the exchange of trade around countries in Africa and also in vulnerable locations.
Thank you very much. Nice talk. Why do you think Ralstonia solanacearum, the model, performed poorly? on Ralstonia solanacearum? Why do you think the model performed poorly on Ralstonia?
Really, like I was thinking, like I look at the relationship between because the Lake Kivu, in the Lake Kivu region we have a gradient of altitude, and I look at the information about the relationship between the altitude and the different weather parameters, and the pattern was a bit different with what we have commonly, but that can be because of the lake. And I also think I think also like the complex cropping system, like with the mixture of many crops and disease, can explain that. But till now, I'm still working on this, and I don't have something really precise. Like, yes, but I just thinking about what can explain it. There is I was thinking about the weather parameters, depending on what we have, and also the mixture of all those culture. Yes. Thank you.
No more questions? So we have to thank you again.
And welcome our next speaker, Safaa KUMARI. His talk, Phytosanitary Management of ICARDA's Germplasm Seed Collections for Better Future Use.
Good afternoon. I will open another chapter from the other hotspot from this region. I'm glad to be here with you today, but I'm very sad to learn what our friend talked in this session. Unfortunately, I attend many meetings, many international conferences, regional conference as well. But for me, this is the first time I see in the program something about the impact of war, about plant pathology. Sorry for that. My name is Safaa KUMARI. I'm originally from Aleppo, Syria. And I'm sure most of you in this room, they know now where is Syria. Because based on very bad story coming from that region, most of the Syrians coming to this region, and sorry for that, but in this session, I will talk about the phytosanitary management of ICARDA germplasm seed collection for future use. Also, most of the, maybe in this room, they know Safaa KUMARI as a plant virologist, but today I will not be a plant virologist. I will talk about something related to germplasm. Because at ICARDA now, I'm working for ICARDA, I'm handling the responsibility of both virology research as well as for the seed health testing. I'm the president of the Arab Society of Plant Protection, and this is another activity now I'm handling with that related to the region. The outline of my presentation will include importance of plant genetic resources, and what is the CGIR and gene bank crisis and how to overcome them, and then what is the rules of germplasm health unit in management of ICARDA germplasm seed collection, and then I will be close my presentation with a few conclusions. Preserving plant genetic resources is crucial for the global food security and sustainability. Plant genetic resources provide valuable trait for developing resilient crop and ensuring the adaptability of our food system to various stresses. Preserving these resources is an investment in a sustainable global future, empowering humanity to overcome challenges and secure abundant harvest for generations to come. More than 750 individual genotypes around the world that collect, store, regenerate, and distribute crop variety and their wild relatives. These gene banks conserve around 7.4 million accessions. 25% of these accessions are unique, while the rest are duplicate. Gene banks are vital to the future of people on Earth, and in sense, they are much more valuable than ordinary bank. Now, if these collections are not maintained, what will be happen? These face the risk being lost forever. Now, just to give you an idea about the CGIAR, maybe some of you are not know what is the CGIAR. CGIAR are the Consortium of International Agriculture Research Center, is a global partnership that unites international organizations engaged in the research about food security. CGIAR research aims to reduce real poverty, increase food security, improve human health and nutrition, and sustainable management of natural resources. It's carried out at 15 international centers operating in 98 countries through a combined them of our 8,000 scientific staff. 11 CG centers out of 15 have gene bank, which conserve around 736,000 accession of cereals, grain, legume, forage, trees, species, root and tuber crop, and banana. And this representing 174 genera and over 1,000 species in 35 collections around the world as a seeds or in vitro material as a living plant in the field or greenhouses. Many of these accessions are crop-wild relative. This is one example about ICARDA gene bank is before the bad time of Syria, because the war in Syria, they started in 2011. The ICARDA gene bank at that time conserved about 134,000 accessions, but now, based on the data of the last year, now the ICARDA conserve 157,000 accessions of barley, wheat, faba bean, chickpea, lentil, grass pea, in addition to this including wild relative, and forage and range plant. Now, seed bank destructions may cause agriculture losses. This is true. But I will give you, before we start, a few examples about the crisis of the gene bank around the world. Gene bank collections have been lost or destroyed due to the conflict of Afghanistan, Burundi, Rwanda, and the Solomon Islands. When gene bank flooded in Thailand in 2011, around 20,000 unique rice accessions were lost forever. The National Gene Bank of the Philippines at Los Banos was damaged by flooding in 2006 and hit again by fire in 2012. The other bad story which is coming to us, which was happening in Ukraine, based on the New Scientist newspaper, they mentioned 10,000 of seed samples have been destroyed from Ukrainian national seed collection during 2022. In a consolidation effort, the collection was relocated this year. The very bad example, this is what happened in my country. And when because as I mentioned, I'm originally from Aleppo, Syria, at ICARDA headquarters is still in Syria, and when the war started, I was at that time in Syria. And when the crisis is coming, the future of one of the world's most important seed collection was put at risk. What followed was an internationally coordinated rescue to ensure the preservation of this priceless heritage. This, when you look for this picture, you will see what happened with the gene bank of ICARDA at that time. This is the pictures. This is a bit before 2011, and this is the building, what happened after the war. This is the seed shelf. This is what happened for the seed shelf of the gene bank. Based on that, we lost everything in Aleppo, the laboratory, gene bank, and the people left outside Syria. Now let's go to how we can save our gene bank or germplasm over the world. I will mention a few examples, but let me bring some water, please. Thank you. Sorry, when I remember Syria, I feel Dr. Kelly Fuller is an American scientist, agricultural scientist, say that at any given time, there is almost always something bad going on in one of the gene banks around the world. Dr. Fuller was a former executive director of the Crop Trust, and during his job, he pushed with the CGIAR stuff, how they can save the seeds for a long time. And then they start to build the Svalbard Gene Bank in Norwich. This is they opened it on February 2008. And I'm sure most of you heard about this place. Now there is more than 1 million seed sample from different gene bank is saved as a duplicate just to save the seed for the next generation. The second example I will see about how we can save our germplasm. Save duplication, what is called now black box. As per the FAO gene bank standard, gene bank should have safe duplication arrangement with one or more institutions, including international, regional, and national gene bank, as well as the Svalbard Gene Bank for it. Accordingly, during the period from 1993 till 2006, ICARDA deposit most of their material in different locations, either with the sister gene bank, like Summit in Mexico, or ICRISAD in India, or with the national gene bank like Ankara in Turkey, or at Svalbard at that time. We save most of our material in different gene bank. Now, after the war, ICARDA, we started to retrieve this material from different locations. We retrieve most of all the material from during the period from 2015 to this year. All the safe duplicate were retrieved from different gene bank, as I mentioned, and then we moved to Lebanon and Morocco, because now we established 2 gene banks, one in Lebanon, one in Morocco, just to multiply the seeds and restock this germplasm. As I mentioned from previous, all this material were lost in Aleppo. Out of 141,000 accessions were at Syria Gene Bank at that time, ICARDA successfully to recover 99%, while only 1,657 accessions lost to the war. If you remember, in the maybe second or third slide, I mentioned about for Philippines, they lost 20,000 unique rice accessions, whereas ICARDA, they lost only 1,600 accessions. I will tell you now in the coming 2 slides why we did that, and maybe in the previous slide I mentioned that where we deposit this material before the war started. Now this is based on my job now. Any seed movement procedure must comply with phytosanitary regulation to enable direct and rapid response for the safe germplasm exchange and retrieval later on. ICARDA exercise maximum effort to maintain the health status of germplasm collection, ensure compliance with the phytosanitary regulation and international germplasm distribution to guarantee minimum losses of genetic resources. CGIR Gene Bank, as I mentioned, we have 11 Gene Bank. They distributed millions of seeds around the world to the farmers, to the researchers, to the breeders. This just to have an idea how many accession or sample they distributed during the period from 2012 to 2020. They distributed 879 samples to 160 countries. 85% of the international germplasm distribution over the world is coming from the CGIR Gene Bank. During 2008 to 2010, we distributed about 500,000 samples, 23 from the Gene Bank and 61% from the breedings material. Seeds have been recognized as an important pathway for the spread of pests and pathway through the human activity of collaboration and distribution. Accordingly, CGIR centers have established germplasm health unit, which is recognized. This GHU is about 11 centers around the world. Each center had gene bank, had germplasm health unit next to the gene bank. To ensure the safety of exchange plant material and complying with the International Plant Protection Convention procedures and international standard phytosanitary measures used by National Plant Protection Organization. The objective of germplasm health unit are avert the spread of quarantine pests in all CGIR germplasm transfer, prevent pest outbreak, and safeguard biodiversity. The International Gene Bank need reports way to ensure that only pest-free seeds are transported across the border. This is exactly our core function, the germplasm health unit. We should test all the material from the collection till the distribution. When the gene bank started to do collection, we should be tested for the seed-borne pathogen during the conservation, multiplication, regeneration, or once you send it outside for the seed duplication or black box, and then to reach the distribution to the other for the breeding material, international nursery, or gene bank collection. We have a very strong protocol. We started from the crop protection practice during the growing season, field inspection, testing, crop rotation for the seed regeneration and multiplication. And when needed, we can use chemical control, seed fumigation, seed treatment, or seed washing. And we tested all seeds, as I mentioned, for all the stages for the seed-borne pathogen before dispatching from ICARDA or from any other CG gene bank against the quarantine regulation for received country. Then dispatch seed along with required quarantine document like phytosanitary certificate, import permit, certificate of origin, and so on. Now, tomorrow seeds, or what is the seed collection for next generation. All the retrieved material which returned to ICARDA were multiplied, tested for seed-borne diseases, and sent it back to the Svalbard for deposit or for storage for long term for the next generation. What is our challenge? We have emerging new diseases and insect pests. Minor pests and diseases are becoming important to the climate change, variable standard and different phytosanitary demand, limited funding, unfortunately, for preventive measures. And recently, during our retrieve the material, we found also we have very difficult procedures when we retrieve the material, especially when we lost the original phytosanitary certificate due to the crisis or due to what happened in Syria. Now, what is the conclusion for my presentation? During the Syrian war, ICARDA lost only, as I mentioned, 1,657 accession. This is an important positive story for collaboration between national and international gene bank working together to protect a unique global resource that we could not have a possible replace if it had been lost. Maintaining plant health during germplasm regeneration, multiplication, and safe duplication is essential to reduce the risk of seed-borne pathogen spread via future germplasm distribution. Germplasm health unit efforts are an important step in providing effective implementation of pest management, which in turn help in maintaining crop sustainability and diversity. Many thanks for the Crop Trust for their support during last 6 years, either through Gene Bank platform or now through the initiative on Gene Bank. Many thanks for our national program in Lebanon and Morocco for helping us to issue the phytosanitary document. And many thanks for my staff in Lebanon and Morocco. And many thanks for you.
We are late. We'll take 1 or 2 questions.
Thank you, Safaa, for excellent presentation. And India is one of the beneficiaries of getting the germplasm from ICARDA. And I want to know, when you send the wheat and all, do you test for viruses and send the material? How do you do that? Do you test only the seeds, or you grow them, grow out, test and send it? Because pea and all lentils we know, but wheat, we really don't get any viruses.
Yeah, ICARDA mandate are legumes, faba bean, lentil, chickpea, and pea, chickpea, lentil, faba bean, and peas. And for cereal, we have wheat and barley. And we have different type of tests, because also we have different type of diseases. We have fungal disease, virus, bacteria, in addition to wheat seeds. And we have different tests. We have list of tests. We have different also diseases. And for each diseases, we have a special test. And if you are interested, I can give you the link. You can go to the ICARDA website and see all the diseases, all the quarantine diseases, and the name of the test which we are using for each disease.
Even though we are late, I have one more question for you, if you want. If you want.
Yeah, no, it's okay.
In your opinion, what are the technical challenges for the germplasm health units right now for you? I'm thinking of tools, for example, for diagnostic. What are you facing as a problem?
No, the problem now is the funding, because now without the funding, special funding, now my colleagues are, they join now, Dr. Lava Kumar, Jan from CIP. All of us now are working as one team. We have one budget. It's coming now, it was coming through Gene Bank platform. Now we have a special activity through the initiative of the Gene Bank. And all of us are working in the same boat. Each one working on different crop. Each one has a different challenges. And at the end, all of us, we have a update technology. We have good laboratories. We have good staff. But at the end, when we reach, we find that maybe the funding is a big challenge. How we can continue. This might maybe our colleagues, they have something else, other challenge. But my feeling is the funding is coming on that one. But also, I mentioned that in the challenge now, maybe this is the emerging diseases. Maybe now we funded AOS when we send it materials for safe duplicate for the other center, and then when the crisis coming and when we lose the shipment document, we'll have problem how we can retrieve this material again. I think now this is maybe Dr. Lava Kumar will be something, our host.
Thank you, Safaa, for putting me on the spot. I would like to add to Safaa that seed health testing, especially for germplasm movement, is most underappreciated work. Most donors don't see it as something to be invested. So as a consequence, it's a big challenge. We require recurrent funding to constantly upgrade our diagnostic tools, the methods. As we encounter new pathogens, we need to automatically change. As we get the new knowledge about the seed transmission of pathogens, we need to upgrade. It requires a decent investment. We are not expecting a huge chunk of money. But even getting funding for routine operations is becoming a challenge as more and more funding is diverted towards the research for innovation kind of work. So as Safaa mentioned, for us the biggest challenge is the funding.
Okay, thank you very much. The last session.
Yeah. And the last presentation will be from Alex Shevchenko from Taras Shevchenko National University of Kyiv. It is also pre-recorded, and it is emerging and re-emerging viral threats for commercial plants in Ukraine, war and other problems.
Good afternoon, ladies and gentlemen. My name is Alex Shevchenko, and I work at the Virology Department of Taras Shevchenko National University of Kyiv, Ukraine. This is an honor for me to be here today at the 12th International Congress of Plant Pathology in Lyon, France, especially given the circumstances. When the war has started a year and a half ago, we were all shocked and no one knew what would come up next. Later on, you start to understand that it may be a long go, and you start writing to friends and colleagues abroad to make some noise, to become heard, to attract attention to this disaster. A friend in need is a friend indeed, as you know, so that's why I'm here today. Before going any further with my talk on emerging and existing plant viral threats in Ukraine, I would like to say my enormous thank you to people from several countries who made this special session possible. Initially, this session has been conceived after communication with Greg Johnson and Malgorzata Andrichka. We have agreed that it would be the right move for the scientific community to know what is going on with plant pathology research in Ukraine and how we cope with this, and that the ICPP platform would be the right place for it. So I use this opportunity to say thank you to Greg Johnson, the immediate past president of the ISPP from Australia, Malgorzata Manka and Malgorzata Andrichka, president and vice president of Polish Phytopathological Society from Poland, and most of all to Didier THARREAU, chair of Scientific Problem Committee of ICPP from Montpellier, France, and to Matthias Stöcke and Nathalie Possereux, ICPP organizing chairpersons from Lyon, France. Without you, this would never happen. From the very beginning, France demonstrated strong support of Ukraine, both diplomatically and practically, for which we are very indebted. I'd also use this opportunity to thank my colleagues, Jean-Jacques Hervé from France and Kateryna UDOVYCHENKO from Ukraine, for doing a great job and chairing this session. Coming closer to the topic of my talk, which I'm afraid will prove to be more social than academic. In short, Ukraine is known for its soils and grain, among other stuff. As you may see, we are ranked number 5 as grain exporter in the world, plus we produce over a half of global supplies of sunflower oil. In 2021, our grain and oil export totaled over $10 billion and heavily dependent on port availability, as this is the preferable option for cheap and long-range transportation of commodities. This is just to give you a brief idea why agriculture is so important to my country. Obviously, being so an agricultural state, you are subjected to plant pathology risks. Indirect estimates show that the total loss of Ukraine from viral infections of plants can reach millions, if not billions of dollars per year. Thus, the control of, or at least understanding, viral infections is crucial for sustainable production and food security. During the last years, we have monitored and studied some viruses of commercially important plants and for the first time characterized new new viruses which are especially dangerous for Ukraine. On the following slides, I will focus on several viruses of major interest which either persist in Ukrainian ecosystems or can be considered emergent pathogens. Economic significance of viral diseases of cereals is hard to overestimate, as yield losses can reach huge figures during epidemics. According to available data, at least a dozen of different viruses circulate in Ukraine. Monitoring of major cereal viruses show that barley yellow dwarf virus, wheat streak mosaic virus, and wheat dwarf virus were the most harmful viral pathogens in Ukraine for years, until now. Interestingly, barley yellow dwarf virus complex has long been considered a major issue with cereals, claiming to infect, say, 50–70% of symptomatic plants in Ukraine. However, for the last 20 years, wheat streak mosaic virus has started, as you can see, to efficiently replace this established dominator, and now accounts for over half of mosaic disease cases in cereals in Ukraine. Wheat streak mosaic virus can naturally infect many cereal crops which are routinely cultivated in Ukraine, including nearly all wheat cultivars, as well as grasses, ensuring survival of the virus off-season. On grain cereals, wheat streak mosaic virus typically causes leaf mosaic and sometimes plant stunting. Wide cereal cultivation in Ukraine, as well as international grain trade, coupled with mechanical transmission and seed transmission of this virus, do not leave much doubt that wheat streak mosaic virus is presumably ubiquitous in Ukraine. Another cereal pathogen, a new guy in the city, High Plains wheat mosaic virus, belonging to Emaroviruses. This is the only known wheat-infecting Emarovirus, originally reported exclusively from the USA, Argentina, Australia, and New Zealand, and now from Ukraine. In Ukraine, this virus has been described as an emergent wheat and maize pathogen back from 2020, and this was also the first communication of this pathogen in Europe. A couple of years ago, we have studied the pattern of infection of these two wheat viruses, wheat streak mosaic virus and High Plains wheat mosaic virus, in symptomatic plants. The results showed that wheat streak mosaic virus was the most common wheat pathogen, as we have discussed, often found in mixed infection with High Plains wheat mosaic virus. Mixed infection is not surprising after all, as both these viruses are transmitted by a single vector, wheat curl mite. I'm afraid we may expect wider spread of High Plains wheat mosaic virus in Ukraine in the coming years, in the regions where the vector is present and susceptible cereals are repeatedly cultivated. Now, raspberry leaf blotch virus is a relative of High Plains wheat mosaic virus and is also an Emarovirus. Initially, this has been reported just 11 years ago from the UK, where it caused leaf blotch disorder in raspberries. In Ukraine, this virus was originally found 5 years ago. Phylogenetically, Ukrainian isolate was shown highly similar to known Finnish and British isolates, suggesting their common origin. In the view of rapid intensification of raspberry production in Ukraine in the last 10 years, coupled with introduction of many new varieties imported from Europe, we suggest that this virus was imported to Ukraine as planting material. Spread of this pathogen in Ukraine remains currently unknown. Apart from cereals, vegetable crops are considered strategically important in Ukraine. Before the war, areas used for production of these crops reached roughly 20% and increased annually. Several highly damaging vegetable viruses have been described. Cucumber mosaic virus, however, is yet another example of the virus which successfully persists in Ukraine for a very long period of time. In Ukraine, cucumber mosaic virus is typically found in cucurbits, tomato, etc. Plants develop varying symptoms like mosaic, chlorosis, mottling, necrosis on leaves and fruits, and so on. Moreover, this virus is often found in mixed infections as well, similarly to wheat streak mosaic virus discussed before. The results suggest long-term persistence of cucumber mosaic virus in the fields, where it may circulate off-season by infecting other available hosts, including both mono and dicots. Just to give you a few examples for Ukraine, recently cucumber mosaic virus has been found in a medicinal plant, Echinacea purpurea, decorative gladiolus plant, and in a brassica weed, garlic mustard, and recently in garlic itself, which is in press now. Speaking about onions, since 2015, large areas in different regions of Ukraine have been massively used for increased cultivations of onions and especially garlic. Apart from domestic varieties, many new cultivars have been imported from abroad, and these resulted in numerous reports of disease from producers. Research identified 2 novel viruses which have not been previously reported in Ukraine: onion yellow dwarf virus and leek yellow stripe virus, both found back in 2019. In onions and garlic, these 2 viruses may induce quite similar symptoms. Typically, however, onion yellow dwarf virus induces leaf mosaic and shoot deformation or plant stunting, whereas yellow stripe virus normally shows up in form of striping or mosaic on leaves. Screening showed that both these viruses are widespread in Ukraine and are often found in mixed infection, leading to more severe symptoms. Average incidence rate were 64% for onion yellow dwarf and about 28% for leek yellow stripe virus. These findings have already placed some additional restrictions on the export of Ukrainian onion and garlic to the EU. Phylogenetic analysis showed that Ukrainian isolates were most homologous to published Chinese isolates, suggesting the recent introduction of this propagating material. To finish with the onion pathogens and to give you a modest insight, last year we've detected many more viruses. Coming soon. Now, the last part of this story. As many potiviruses, turnip mosaic virus has an extremely wide host range. For domesticated plants, this pathogen is considered one of the most damaging and economically important viruses. It was first described in Ukraine in 2018 and hence should have been regarded as an emerging pathogen. However, an impressive number of positive samples and a wide range of infected species indicated that the virus presumably persisted here for quite a long time, which is not surprising after all. In Europe, turnip mosaic virus was reported from nearly every country. Despite Ukraine's strategic geographical location and wide cultivation of different brassica crops for centuries, the pathogen somehow has evaded detection until recently. Screening of commercial fields showed surprisingly high incidence rate reaching over 40%. Among the most typical symptoms on cabbage plants, for instance, were vein binding and leaf deformation. Notably, 22% of infected plants were also infected by cucumber mosaic virus, suggesting high percentage of mixed infection. We also screened urban locations in Kyiv, the capital of Ukraine, where there is no agricultural activity. Surprisingly, these were characterized by equivalently high incidence rate for symptomatic plants. Evidently, associated crops and wild plants may play an important role as reservoirs for the conservation and transmission of the virus in urban areas. The obtained results clearly show that the significant spread of turnip mosaic virus is evident in both agricultural and urban ecosystems. Now, interestingly, sequenced Ukrainian isolate was a recombinant one, homologous to Turkish isolate described 10 years earlier. This suggests that international trade of vegetables may play a role in virus spread, as we used to import a lot of wax from Turkey. As we have discovered for turnip mosaic virus, pathogen evolution does deeply depend on our own activity. In this case, historical trade routes were and remain the factor governing virus adaptation. This is all you need to know about urbanization and globalization in the world of viruses. And this slide shows you a short summary of new viruses and new virus hosts which we have recently described in Ukraine. More news is coming soon as we seem to host plenty of viruses. Ukraine showcases that our ignorance is probably the major factor favoring the emergence and persistence of virus infections. Aristotle wording shown here is no understatement. Apart from biological properties like several means of transmission and extreme adaptability of viruses, there are also other important factors to consider, including non-observance of agricultural practices like crop rotation, absence of regular screening of crops for viral diseases, uncontrolled import of seed or plant material, and wide susceptibility of used cultivars to certain viruses. So we have what we have, basically. Coming to the well-known triad of One Health, it is important to remember that it's true not only for us humans whose well-being depends on plants or animals' health. It's also working the other way around when our activity does shape the ecosystems and define their future. We're all connected. Now, less pleasant part of my talk will be about the worst side of globalization, the conflicts and their consequences. By the time we will be watching this video, it will be nearly a year and a half of war in Ukraine. To be more precise, 543 days. Is it much? It's impossible and painful to account for all the losses we are continuing to bear. I will not be telling you about the casualties, illegal deprivation of kids or occupation. You've seen it all on the news. But I will show you some figures directly related to agriculture and crop production losses. Some of these you may have seen today already. We will see. To start with, millions of tons of grain have been stolen. Huge number of elevators have been destroyed, and large areas of farmland with crops were either burned or become unusable in some way. So in addition, before the grain deal and after its stoppage in late July, our Black Sea ports remained blocked, preventing grain export. This has already resulted in food shortage seen in many countries, especially in Africa, as you may have heard from a nice talk earlier in this session. Direct damages are relatively easy to measure. Lost or damaged equipment, facilities, crops, and so on, all easily countable. Mostly these come from stolen or damaged machinery and grain. Grain itself was nearly $2 billion worth. Total anticipated direct damages amount to nearly $9 billion. See that these are based on March figures, so the actual numbers will be higher by now. Just to give you a recent example, Black Sea grain deal was stopped by Russia in late July. Since then, 180,000 tons of grain were destroyed in Odessa region just in 9 days, plus 26 infrastructure objects in several ports, plus 5 civil ships. And this is continuing right now. Just a short glimpse into what is going on. Even though the war itself does not seem to have direct effects on plant health, the indirect consequences are certainly huge and long-term. A lot of land was either occupied or made unusable because of heavy mining, soil contamination, and so on. Obviously, our producers had to take measures to relocate businesses, switch crops, introduce technological or logistic changes. All these factors amount to higher production costs and lower crop production. As you may see, such indirect losses may amount to way over 30 billion. The figures may not be that accurate as direct damages, but still remain staggering. This is in agreement with FAO assessment predicting over 30% decrease in cereal production this year and confirming that many farmers change preferences from cereals to other, more profitable crops in the view of problematic export from Ukraine. Luckily, our producers have just reported a record high yield of winter crops in most of the regions. Another example of long-term and nearly uncountable losses, the destruction of the Kakhovka Dam 2 months ago. In addition to humanitarian disaster, this has led to flooding and washing away crops growing downstream of the dam, whereas later on the region became unusable. One of the main regions producing vegetables was made a desert, a contaminated desert. So what do we have in essence? A lot of land became unusable, so we have to switch regions, crops, and technologies and to use land more intensely. Obvious consequences here are disruption of global supply chains and much higher production costs. Can this lead to soil exhaustion and the emergence of new pathogens? The time will show. We will be watching. Most importantly, we lose people. Over 5 million of people were relocated in Ukraine, whereas over 6 million of Ukrainians sought refuge in other countries. And 20% of them say they don't intend coming back when it's over. So this is massive brain drain. We lose a whole generation and we lose professionals who would help rebuilding the country otherwise. The last but not least, the estimates show that we need nearly $30 billion for reconstruction of Ukraine in 10 years' time. That's roughly $3 billion per year. such are the numbers. The good news is that it's already started. The example of which I'm fond of is the Resilience Bursary established by the International Society for Plant Pathology. So far, a dozen of Ukrainian plant pathologists has already benefited from this fund and took advantage of not only of the refuge in Poland, but also of professional development. Polish Phytopathological Society worked closely with ICPP and developed a framework for selecting candidates, finding placements, arranging childcare and accommodation. In my view, it is vital to keep professionals in business, especially when you plan to rebuild your country soon after the war is over. And this brings me to more thank you to people who spend a lot of their time and lives to support my colleagues, even at the cost of their birthday party. I would like to thank John Leach, Greg Johnson, and Matthew Sparrett from the ISPP, and again, Malgorzata Manka and Malgorzata Andrichka from Polish Phytopathological Society, who made every effort to make sure that my countrymen may feel needed. Okay, this is it. My time is up. At this point, I would like to say my thank you to my colleagues from Ukraine, Irena, Tatiana, Halina, and Ilya, who really helped me a lot with collating this data. And here is my contact info. Please feel free to mail me if you have any questions or interested in working with us. With this being said, I would like to wish you very productive time during the Congress, as well as interesting talks and future collaborations. Please support Ukraine, support our people abroad, especially their professional pursuits, and also please encourage them to come back when it's safe, to bring best experience and skills to rebuild our country. Thank you very much for your attention and the opportunity to talk here today. Slava Ukraini!
So it's time to conclude this session and to release you for the coffee break. So we just want to
thank you for your interest to this session, to this topic, very important, and to thank you for your attention. I wish you good time to continue experience at Congress. Thank you.
And thanks to the speakers also.